So, my name's Mark Pallen. I used to work here in Birmingham for 12 years. And a few years ago I got headhunted and moved to the University of Warwick. I've regretted it ever since. And I come back here every now and then to give lectures and reminisce about my time in Birmingham. I'm about to start a new job in Norwich in a few weeks' time. But I do like to come back here every year and give these lectures on human evolution. One of the problems, though, is that they keep putting me in the timetable for 3 lectures on 3 different days. I keep saying, I don't work here, I don't want to come for 3 separate days. So this year I managed to badger them into giving me 1 2-hour slot, and I'm going to scrunch the 3 lectures into that. Now that sounds bad, and it is bad in a way, because it means at worst I could just have a monologue for 2 hours and try and scrunch 3 lectures into 2 hours. But what I'm going to do is actually avoid that by having some breaks. So what we're going to do is we're going to cover 3 topics. The first one is becoming human. What makes us human? And what's our closest relative in nature? And what has happened to us since we separated from the most recent common ancestor from that relative? Then we're going to have a musical interlude just to summarise things up till then. Then we'll move on to this, and we'll have a comfort break. Maybe we can go and get some coffee. I think there is a coffee shop just out here, actually, so it's quite conveniently placed. And then after 5 or 10 minutes, we come back, and then we look at the second part, which is going to be called the Scatterlings of Africa, and it's going to look at the origins of anatomically modern humans and the story that we all come from Africa. So everyone in this room is an African. Even if you don't have recent ancestry in the last few hundred years, you have recent ancestry in the last few tens of thousands of years in Africa. And then what I'm going to do is something that you probably won't have come across in any of your courses, which is we're going to summarise that part of the course in a rap video that will summarise everything we've learned in that 40 minutes or so talking about the Scatterlings of Africa. And then we'll have another break, and then the final part will be looking at ancient DNA and what we've learned from ancient DNA about human evolution. Okay, so that's the introduction. So if we want to find out about human evolution, where can we go for information? So when we look at us, anatomically modern humans, one of the things we can do is we can look at population genetics. We can look at sequences from mitochondria, from Y chromosomes, from what we call autosomal genes on the other chromosomes. We can actually now, in the last few years, even look at whole genomes and analyse whole genomes. And that can give us some idea about how we came to be where we are, how the world got peopled by people, the various ancient migration routes and so forth. We can go back in time now and we can look at extinct hominins. So hominins is the name given to that branch that's branched off, our branch that's branched off since the most recent common ancestor with chimpanzee, with our closest living relative. And we have some extinct examples of that particular clade, and we can look at the fossil record and we can look at the morphology of bones and we can make assumptions about relationships based on that. But as we'll see during the course of these talks, we now have ancient DNA as another way of looking at things. And just in the last couple of years, we've even been able to use modern genomes to fish out information about extinct hominins. And then, of course, we can go and look at living primates. And we can do the same kinds of things as we can do on anatomically modern humans. We can look at phylogenetics, we can look at genomic studies. We can look at morphology, but actually now morphology seems to be the poor cousin. We get so much more information out of DNA sequences that this has now become the preferred way to try and work out what's related to what and who's related to whom. So let's start with this first session on becoming human. And we have to first start off with an assumption, like where do we come from? What are our closest relatives? Well, it was fairly obvious. I've chopped out some things because I didn't want to take up too much time. But you know, even to people like Darwin and Huxley, it was fairly obvious that our closest relatives were the great apes, and particularly the African great apes, so chimpanzees and gorillas. And if you look at, just look at them, isn't it fairly obvious, though, how the relationships work, that gorillas and chimps, these brutish creatures here, are obviously more closely related to each other than either of them is to this beautiful Madonna and child, to modern humans. And there are a lot of reasons when you first look at it that make you think that way. So gorillas and chimps have knuckle walking. We stand upright, they walk on their knuckles. We have all these distinctive features, bipedalism, giant brains, we're hairless, tools, culture, language. And so a few decades ago, when people looked at this, the only thing they said was, well, did the humans branch off? They must have branched off a long time ago, and humans must have had this distinguished lineage that goes on for many millions of years. And the only question was, did we branch off before the orangutans or after? The thing was, there were no chimp fossils, and so that didn't really help. But this was the working assumption, that this is the way that the relationships look. Chimps and gorillas obviously look closer together, and humans branching off early. But in fact, the first threat to this comfortable view came in the 1960s with a seminal paper from Sarich and Wilson, where they used immunology. So they basically raised antibodies to various proteins. And what they said was, actually, if you look at the relationships between humans and old world monkeys, and you look at the distance in terms of reactivity of antibodies, and you assume that they were about 30 million years ago, the common ancestor with us and old world monkeys, then we and apes share a common ancestor only 5 million years ago. So it's basically like it's 1/6 of the distance to monkeys, the distance to the apes. And you know, people at that time were talking about 10 or 15 million years of unique human evolution. And this was a real threat. Oh, hang on, can it really be that recent that we had a common ancestor with apes? The thing that actually made us really move forward in this area, though, was the use of sequencing and sequence-based phylogenies. So we can take a bunch of DNA sequences, and people started off doing this on single genes. Now we do it on whole genomes, and you can just line them up. So you can look at what we might call homologous sequences, the regions of homology in these sequences, and you can see, in most cases, they're the same. But every now and then there's a variant. So you can see there are 3 of them have an A, but the orangutan's got a C, which might make us suggest that the orangutan is outgroup and the other 3 are more closely related to each other. And then over here you've got human and gorilla and orangutan got a C, but the chimpanzee's got a G, which is a bit weird, it doesn't quite match. And this is the problem, is that you do get mutations happening all the time, and some of the time, well, most of the time they do agree with each other, but some of the time you do get ones that actually confound your picture. But when you do this kind of thing, you get a tree where humans and chimps are more closely related to each other than either of them is to gorillas, and the 3 African apes, us and those 2, are actually more closely related to each other than they are to the orangutan. In fact, it was clear that the way to conceptualize this was a trichotomy, that we had these 3 species, humans, chimps and gorillas, and we had to keep an open mind as to what the relationships were, and not just say, well, these are dark and brutish and hairy, and therefore they must be more closely related, and actually look with other eyes, if you like. So the first attempts at using DNA sequence data actually gave us kind of contradictory results. It wasn't a clear answer. But, and here, this is from an old review from 1997, basically pulling out the various data sets to do with different genes. And you can see that as time went on, by the time we got to the late 1990s, we basically had many more data sets that were supporting a clade where humans and chimpanzees were together than were supporting the idea that chimps and gorillas were more closely related, or that humans and gorillas were more closely related. And if you just took all of the sequences you could find and you concatenate them together and drew a big tree based on that, it tended to support the idea of a human-chimp clade. Now the question is, why is it so confusing? Why is it difficult like this? And the reason is that some sequences are incongruent with the basic species tree because they have different evolutionary trajectories. So you might have a couple of variants of a gene. So you've got two variants here of gene B, and two of them find their way into species 1 and 2. One of the variants finds its way into those, but the other variant, that variant dies out in species 3, and instead you get the survival of this other variant. So think about it this way, that if we looked at things like ABO blood groups, which we know actually that polymorphism goes all the way back to chimps and gorillas. If we chose this lady here and she turned out to have an A, and then we chose a random gorilla that also had an A, and then we chose a chimp that was B in their blood group, we might draw false comparisons from just looking at these small data sets and looking at small numbers of genes. And so you have to actually look at the whole picture, all of the genes, or as many genes as you can, to get a proper consensus view. So before we started sequencing genomes, which we began about 15 years ago, we started getting these first genomes, we had these various lines of evidence, immunology, as I mentioned, from Savage and Wilson, protein sequences, chromosomes, actually counting the numbers of chromosomes and so forth, and looking at the arrangements on them, DNA hybridization sequences, gene expression profiles. And basically the consensus was coming that humans and chimps were most closely related. When you look at morphology, it's a little bit harder. If you look at general morphology, some of the original papers were saying, well, actually, no, chimps and gorillas do look more like each other than either looks like a human. But if you look at specific bones, like temporal bones, and there's another study done on soft tissue, again, these support the idea of humans and chimps coming together. Anyway, that's a bit of history, because now we have genome sequences from all of these different species. From humans, from chimps, from gorillas, from orangutans. We even have monkey sequences from rhesus macaque. And when we've looked at these, we now come to a clear conclusion that humans and chimps, despite these superficial features telling us otherwise, humans and chimps are more closely related to each other than either of them is to the gorilla. So if we look at the tree, the gorillas branched out first, and then the chimps and humans split. And in fact, when the gorilla genome was completed, it basically confirmed this. And as it shows you here, you had, if you look up there at figure 1a, it says phylogeny of the great ape family showing speciation of human, chimpanzee, gorilla, and orangutan. And the horizontal lines indicate speciation times. So there was only a short distance between the time when we split from the gorillas and the time we split from the chimpanzees, so very closely related compared to the orangutans. And then the interior grey lines here show you incomplete lineage sorting at a particular locus, which means that kind of explains why we get these confusing results with certain genes but not others. In fact, there are more than one species of chimpanzee. There are these other species that we now call bonobos. They used to be called pygmy chimps. People want to give them the recognition they deserve of being a completely separate species. These are sometimes called the sexy chimps because
They actually engage in all sorts of sexual practices. And in some ways, people say they're more like humans than the common chimpanzee. Common chimpanzees have all these dominance hierarchies and all that sort of stuff, whereas the way things work in the bonobo society is they all have sex with each other to get along. And in fact, here's an example even of female bonobos having sex with each other, rubbing their genitals up against each other as a way of just chilling out and getting along with each other. More recently, we've had sequences from bonobos as well, and we can actually start to pick apart the relationships between common chimps and bonobos and humans. And again, we can see 95% of sequences here conclude that actually bonobos and chimps separated from each other after their lineage separated from humans. So we're not more closely related to bonobos than we are to chimps, or to chimps than we are to bonobos. And according to the estimates on this paper, when you look at different papers, there's a slight variation. They put the split of humans and the chimp lineage about 4.5 million years ago, and then they put the split between bonobos and chimps about 1 million years ago. But the thing the number most people bang about is 6 million, 5, 6 million, 4.5, probably is a bit decent. But the key point is that we're not 6 million years away from a chimp. And this is a key point. We're actually 12 million years away from a chimp, because both chimps, the chimp lineage and the human lineage, have both been evolving. Things didn't just stop after the chimps went one way and we went the other way on their lineage. They evolved, we kind of clearly evolved and became something else. But so did they. And actually, this is another example of misleading thinking in evolutionary terms. And a few years ago, this was all these assumptions were challenged when people started to realise actually bipedalism is actually a fairly ancient trait of our lineage. So this idea that the great apes, the African great apes, chimps and gorillas, do knuckle walking, and therefore the most recent common ancestor must have been a knuckle walker as well. That's been challenged now, particularly by the finding of this fossil species, Ardipithecus ramidus, which only is very close, just a million years, 1.5 million years away from the common ancestor, and actually had upright walking. And they pointed out in the Nature, science paper, that there's a series of science papers on this, that despite the genetic similarities of living humans and chimps, the ancestor we last shared probably differed substantially from any extant African ape. And so they've become quite different. They've actually, independently probably, gorillas and chimps, learnt how to knuckle walk, and the ancestor may well have been bipedal. And Susanna Thorpe here in Birmingham has actually been doing research over the last decade or more, showing that actually even if you go to orangutans, you can see that they stand up in the trees, what's called arboreal bipedalism, where they'll walk along branches standing upright. So that was an interesting challenge to realise that actually the most recent common ancestor may have been more human-like in many ways than chimp-like. Okay, so we've established that we're most closely related to chimps. And that's kind of interesting in terms of the branching patterns and relationships, family relationships, if you like. But the key question really is, what makes us human? Because chimps, in the vernacular, they are animals. They are different from us. We're human. What actually happened to us on our lineage to make us what we are? Well, the answer is there is no easy answer. One of the things that has been realised now for a few decades is that most evolutionary change, when you look at sequences, is actually neutral. So although you can see the steady accumulation of mutations in a lineage as time goes on, many of those mutations don't have any readout in terms of a change in the biology of the organism. So if you remember that the genetic code has this redundancy, and most codons actually have some ambiguity at the third position. Doesn't matter whether there's a G there or a C there or an A or a T, doesn't matter. And so you can get all that kind of random variation going on without making any difference. So trying to find out what are the adaptive changes which actually make a difference to biology and which have been selected for by evolution, by natural selection over time, that's a hard ask. There's a lot of noise and a small signal. The other thing is that there's no simple linear relationship between morphological change and sequence variation. So if we look at the differences between a human and a chimp, these are similar to the differences, these are much greater than the differences that we'd see between two mice that have from different species. The mice all look like mice, we look like humans, the great apes look like apes. But if you look at dogs, all the variation between a Great Dane and a Chihuahua is squeezed into at least a tenth of the variation, if not greater than that, between us within human populations. Oh, actually, so in the Venn diagram there, it shows you a few of the things that have been put forward as potential changes that might have made a difference in our evolution. So protein evolution, so actually changing the amino acid sequences of proteins which changes their function, that's one option. Missense and nonsense mutations that truncate proteins or stop them even being made functionally. Small indels, small insertions or deletions. Duplications of proteins so that we end up, or genes, so that we end up with more of the protein being made. Substitutions in the promoter region, so we get upregulation or downregulation of genes, or that they get rewired, so they're regulated in a different way in response to different signals and different regulators. And of course we can get deletions as well. So we can get loss of exons as one potential way. So just looking in the broadest terms, we can look at changes in chromosome structure between humans and chimpanzees. And the most obvious difference is there is a difference in the number of chromosomes. So human chromosome 2 is actually represented by 2 chromosomes in the chimpanzee. In fact, I think the chimpanzee is the ancestral state, and 2 chromosomes have merged fused in our lineage to give us this chromosome 2. That probably means that you're not going to see a humanzee. If any of you wanted to go and mate with a chimpanzee, you're not going to get viable offspring because of this chromosome mismatch. People have done experiments, the Soviets did experiments on this sort of thing, but nobody's actually made it work. There are duplications, segments get duplicated and moved around, you get inversions, translocations, and so forth. If we look at single nucleotide substitutions, we've got about 1.23% of these substitutions occurring. That's the kind of rate we're seeing. And if we look at proteins that you have, that have obvious homologs in the chimp, homologous proteins, usually there's only 2 amino acids different. Doesn't mean that you've evolved on our lineage 2 amino acids different from the chimp. The chimp's evolved one lineage, 1 amino acid difference from the common ancestor, and so have we. But in fact, 29%, nearly getting on for a third of the proteins that you have and the chimp have are actually identical in sequence. There are differences also in these repertoires of what we call endogenous retroviruses. So all our genomes are littered with the remains of viruses that have invaded the genome and then hunkered down and actually, in many cases, stopped being active as viruses. They're just left as sequences in there. Similarly, there are these repeats, the so-called ALU repeats, which cut and paste themselves out from parts of the genome and duplicate themselves. So that's why most of our genome is just junk. It's just this repetitive stuff. Seems to be much more of that going on in the human lineage than in the chimpanzee lineage. One chromosome that's been looked at in detail is the Y chromosome, and it appears that this is an area where there has been a huge amount of evolution, particularly on the human side. But also, if you look at the chromosome 21, for example, you can draw on this chart here, you've got a straight line there. Everything is collinear on chromosome 21 between the human and the chimp. Whereas when we look at the Y chromosome, there are all sorts of rearrangements where things are flipped round, so you get the diagonal in the opposite direction there, all kinds of rearrangements going on. And it says here that the extraordinary divergence was driven by 4 synergistic factors. The prominent role of so-called MSY, which is the male-specific region of the Y chromosome, the part that actually makes males males and is responsible for kickstarting testosterone and all these other things. Hitchhiking effects, because there's no crossover going on, so if you get a mutation associated with another mutation that's beneficial, the mutation, even though it's not beneficial, just hitchhikes on the back of the beneficial one. And also it says that there may be these other changes, these differences in species mating behaviour, so that particularly among common chimps, there's much more male dominance, their testes are larger, there's much more of this kind of competition going on, and maybe that's what's responsible for this. Now, one of the interesting things that was discovered when they first sequenced the chimp genome was that some of the genes in the chimp look like disease states in humans. So in humans, for decades before, we've been chipping away, looking at the genetic basis of disease and actually working out which are the variants in a particular gene that are associated with the disease state in humans. But actually, when they looked at chimps, they found that many of these, you can see nearly a couple of dozen of them here, where what the chimp has, we would call the disease state in humans. So this is a bit weird, slightly odd. In some cases, they could resolve that the ancestral state actually looked like what we would now consider the disease state. So what this suggests, again, things are not simple. Things don't just, one gene varies in isolation, its function changes in isolation. All of these genes are working in networks of other genes interacting with each other. And what must be happening is that there must be compensatory mutations going on in other members of those networks that mean that in a chimp, that gene functions and has its role, whereas because those compensatory things are not happening in humans, we get the disease state. So it's a puzzle. Now this is, sorry that I should perhaps put this onto 2 slides. I just grabbed this out of a recent review from 2015, where they're trying to actually add up all of the things where we can point at differences in the sequence that might actually account for differences in biology. And I'm not going to go through them all, actually. There's just a few of them listed there. I'm going to focus just on 2 of them as examples, and then I'll give you a link. If you want to find out more, you can. So one of the genes that's actually been most focused on is a gene called FOXP2. So this was identified in humans as a Mendelian dominant mutation in one particular human family, it's given the initials KE, where the family had difficulties in speaking. And what they found was that there was a missense mutation in a domain, so-called forkhead domain, of this FOXP2. It turns out that FOXP2 is actually a regulatory protein, and it regulates, I think, 200 or 300 genes in the brain. It quickly got given, particularly in press releases and stuff like that, got given the name of the language gene. But of course, that is very simplistic thinking, that language doesn't just come from one gene. There are probably many hundreds, if not thousands, of genes that all impact on our ability to speak. And it doesn't only function in language, it's also involved in motor coordination. And what people found when they looked at what happened when you have this mutation is you've got changes in the basal ganglia, and it's shown that in the embryological studies that actually FOXP2 does actually modulate the development of basal ganglia. Interestingly, when people looked in birds and in bats, they found that FOXP2 also seemed to be implicated in making sound, in bird song, and in bat echolocation. But as a convergent evolution, it wasn't the same mutations that you would get in humans. But what tripped interest in this, from the evolutionary point of view, was when people looked at the sequence variation in FOXP2. And what you find here, this is showing you, so we have, as I hinted at earlier, there are two kinds of mutations in protein coding genes. You have synonymous mutations and non-synonymous mutations. Synonymous mutations where the amino acid doesn't change, the nucleotide sequence changes, but it doesn't change the amino acid sequence. And non-synonymous mutations, the ones we're interested in, where they might have a biological readout. And what we've got here, we've got non-synonymous mutations first, and then the number of synonymous mutations second in these numbers. And you can see that if we go back over time, back to the common ancestor, we can see that from our common ancestor with a mouse, on the mouse lineage there's just been one change in amino acids and 131 synonymous changes, just kind of noise, evidence of mutations going on. And similarly along the rest of the lineage here, we see many more synonymous mutations and non-synonymous until you get to the branch leading from humans to humans from the most recent common ancestors with chimpanzees. And then suddenly we get two non-synonymous mutations and no synonymous changes. So we've got two changes in the codons in this gene between us and chimpanzees, and both of them actually are changing an amino acid. So that was found to be, that was looked upon as clear evidence that this was some kind of smoking gun, that this must be involved somehow in language production and maybe in evolution of language production. And so people did some interesting experiments. They knocked out FOXP2 in mice, and they showed that you got developmental delays, and particularly defects in communication, in ultrasonic communication. They also then knocked in the human, they knocked in the mutation that you get in this human family and showed that they also had developmental problems in mice. But then they kind of did the Doom-Watch experiment, where they took the human gene and they put it into a mouse. They humanized the mouse by putting in the human version of FOXP2. They were generally healthy. They had some slight changes in their vocalization. They had slight changes in their behavior. They had to actually look very carefully at these mice because there was always this concern they might suddenly turn into Mickey Mouse and start talking. You know, that was the worry in the background. That would have been a very remarkable result if just one gene had made such changes, but they did have to look at this. But they did find that there were appreciable changes. Now, it's unclear what this all means. As I say, it's ridiculously reductionist to say that this one gene is the language gene and these changes are in some way single-handedly or even predominantly responsible for our ability to speak. But it is an interesting first kind of handle on this particular problem. One more gene I mentioned in passing, because time is moving on, is AMI1, which encodes amylase. So you probably remember from school days when you did those experiments and detected salivary amylase and showed that when you applied that to potatoes, you could show that starch was being broken down. And there are multiple copies of amylase in mammalian genomes, but there appears to be a massive increase in the gene dosage and the number of different copies of the amylase gene in humans. And this actually correlates with the actual abundance of amylase in saliva. So the more genes you have, copies of the gene you have, the more amylase you produce. And it's been shown when you look at world populations across the world, populations that have a high starch diet tend to have more amylase copies than those that have low starch diets. And this raises interesting questions about the human evolutionary trajectory. We have become much more gracile in terms of our jaws and teeth and so forth over time. One potential answer to why that should be the case is that we actually invented cooking. And cooking food actually helped, and there's a book which I actually just just decided to buy a copy on Amazon actually a couple of days ago, hasn't arrived yet, called Catching Fire, where this guy actually argues that cooking was a serious influence on human evolution. Now, people always used to say, oh, well, it was cooking that gave us access to meat, and that made our brains grow and all that. But that may not be the case, because cooking tubers, things like potatoes or carrots and so forth, that also liberates huge amounts of calories. And so cooking, together with this massive increase in the amount of amylase, may well have had an adaptive role in human evolution. If you can get access to glucose from these polysaccharides very quickly, you can easily see that this might be an evolutionary advantage. If someone's actually got severe diarrhea, even if they can scavenge a little glucose from their diet, that may tip the balance in whether they survive or not. Now, I've skipped over quite a lot of stuff. I could have spoke for 2 hours just about all of these changes in the human lineage and the way in which they're being picked apart. This is I put a review into the folder I've shared with you. This is an interesting summary of where we're going with this, an area of extreme interest at the moment, and extreme excitement because there are so many potential openings here. So basically you can take cells from each of these different primate species, you can go in and you can reprogram them, and so you can make stem cells perhaps from them, you can grow up organoids, you can go in and do genome editing, so you could take a human cell and you could give it the chimpanzee version of a gene. You could look at RNA-seq and measure gene expression in different tissues. You can look at epigenetic profiling. And with all of these approaches going together, where we can actually experimentally investigate the effects of mutations at the molecular level, the cellular level, and at the level of tissues and organs, I think during your working lifetimes, we probably are going to crack this issue of what it was that made us human. What were those changes? We're still a long way from that at the moment. But one way of thinking about this is that we actually have the information because we know all those differences between the genome of a human and a chimp, but we just don't know which ones are the important ones. And it's just a matter of sieving through millions of pieces of data to find the stuff that is actually important. And the answer must be out there. We are different from chimps. There must be something out there. Now, before I finish this part, it's worth just pausing, though, to consider, as did the people when they wrote the first chimpanzee genome paper, that there is a big problem in terms of population sizes. So human populations, over 7 billion of us, but many of these other great ape species are now endangered, some critically endangered. So there are more students here at the University of Birmingham than there are individuals within the eastern gorilla species or in the Sumatran orangutan. So this is a serious problem. Another issue that's been raised when you realise that there's only this razor-thin difference between us and chimps is, do they have rights? Should they be granted personhood because they're so similar to us? And that's a lively discussion. And it has actually reached the courts in a couple of jurisdictions as to whether great apes do have personhood under the law. Now, I've skated over the surface here. I've shown you the tip of the iceberg of what we could talk about. And just while preparing this lecture for the first time this year, I actually came across a site which actually is really great. It actually collates all the information that's out there. It's called so-called matrix of comparative anthropogeny, which is a word that they resurrected. It's an old word that hadn't been used for several decades, but basically the idea of making humans, becoming human. And they've got these couple of links here that actually show you information. They catalogue basically all of the genes that might have a role,
and they look at the evidence and they catalogue the evidence as, yes, secure evidence or possible or weak evidence and so forth. Oh, I keep trying to push the wrong button. There we go. So now we're going to have a musical break and then have a 5-minute break. So this is a song which summarises anything about
A man named Darwin had a revelation. He said all life evolved from the confusion. He had a theory. It made some people nervous when he came to the embarrassing conclusion. We come from monkeys, we're not so special. We've only got a little extra DNA. The chimpanzees are very close to us. Australopithecus, he's not so far away. We come from monkeys swinging in the trees. A little bit of monkey inside you and me. A monkey in the middle, a monkey on your back. It's a fundamental problem, but a scientific fact. Come from monkeys. Feeble-minded said creation was the answer. They got upset, their nose got out of joint. They say we're missing the monkey in the middle. They're picking it kinda proves the point. You're not so hairy, you haven't got a tail. You're not adapted for living in a tree. But if you dare to count up all your chromosomes, you're 98.4% a chimpanzee. We come from monkeys swinging in the trees. A little bit of monkey inside you and me. A monkey in the middle, a monkey on your back. It's a fundamental problem, but a scientific fact. We come from monkeys. Monkey bars feel the primal beat. 6 million years we've been walking on our feet. A barrel full of monkeys swinging from the family tree. We don't want to start a social revolution. We just want to talk about some monkey evolution. But the knuckle dragon stool flipping fools just don't want to see.
And of course, knuckle dragon is the right.
Of evolution, you might just call it intelligent design. But if the universe keeps on evolving, then maybe you should just evolve a better mind. Scatterlings and fugitives, hooded eyes and weary clouds, seek refuge in the night. They are the scatterlings of Africa, each uprooted one, on the road to Elamanga, where the world began. I love the scatterlings of Africa.
So this is a track from a guy called Jonny Clegg, who is an anthropologist. He also wanted to be the first white Zulu. He actually went for an induction, became a Zulu. And back in the days of the old apartheid regime, he was a powerful voice, cultural voice, that was against the kind of determinism that they had of black people being black people, white people being white people, and never the trace of me. So, the second part of the talk is going to be about the so-called out of Africa hypothesis. So, where does this idea come from? Well, the inklings of it come from actually Charles Darwin himself. When he wrote The Origin of Species, he was trying to get through things quickly and perhaps trying to avoid controversy. He didn't say much about human evolution, just said light would be thrown on the origin of man and his history. But a few years later, he then wrote The Descent of Man, and he speculated that humans originated in Africa with this particular statement here, that if you look around the world and you look at where extinct animals are and their closest living relatives, they're usually in the same place. And if you look at humans and you look at our closest relatives and the area that where we probably had these extinct ancestors with those species, it turned out that that was going to be in Africa, more probably, he said. But after Darwin, the focus moved away from Africa. People started turning archaic fossil human lineages outside of Africa, so the so-called Java man and Peking man, and in Europe as well. That was in Asia. In Europe, we got Neanderthals and so-called Heidelberg man, and then there was this famous controversy, a fraud, in fact, of Piltdown man based in England, I think in Kent. And this was a problem, largely of conception. I mean, there was obviously these fossils being found, but also the idea that, well, you know, humans outside of Africa seem to be in a much better state, and Africa can't really be where we originated from. And there were these different theories, so-called multi-regionalism, where the ancestral species Homo erectus turned into Homo sapiens kind of everywhere, across the whole world, and just different local, regional features stayed there, but one species kind of magically turned into another. Others said, oh, there must be this long pre-sapiens lineage that we don't see, that's separate from the archaic forms. And others said, oh, maybe there was an ancestor with Neanderthals. But in recent decades, Africa has come back as the focus of the cradle of humanity. And the evidence here is that we are finding the earliest hominid fossils are found in Africa. The earliest Homo erectus fossils are from Africa. But there are actually very early, 1.8 million years ago, hominin fossils from outside of Africa, particularly from Georgia, from Java. And so it's clear that if the human lineage did originate in Africa, when we split off from the chimpanzees, fairly early on, not much off 2 million years ago, there was this out of Africa migration, sometimes called out of Africa 1. But the question also then focuses on what about our actual species, Homo sapiens? Where did that originate? Where did we originate as a species, and how? And how did we come to be distributed around the world? Now, put this up just again to show you that science is not always free of culture, that there's racist thinking in science back in the day. There was a guy called Kuhn, an unlikely name for an anthropologist, but that's in the 1960s. He said that with these 5 separate lineages that all evolved in parallel with Homo erectus, and that if Africa was the cradle of mankind, it was only an indifferent kindergarten. And other kind of similar hypotheses, spectrum hypotheses, Neanderthaloid phase, and so forth. Very kind of what we would now see as very primitive and actually biased thinking. And it was in the 1970s that things started to crystallise down into 2 competing theories. So one called the recent African origin, or out of Africa hypothesis, popularised by the English paleoanthropologist Chris Stringer, who said, looking at skulls and the morphology of skulls, that Homo erectus evolved into Homo sapiens in Africa, and then only recently, in less than 200,000 years ago, did humans actually come out of Africa and disperse around the world, and they would have replaced any existing populations in around the world as they moved out. So if there were Homo erectus in Asia, there were Homo neanderthalensis in Europe, they got replaced by modern humans.
An alternative view that was championed by American anthropologist Milford Wolpoff said that actually no, Homo erectus evolved into modern humans in different locations throughout the world, and there was continuity of gene flow. And so maybe the Homo erectus that you see in China, maybe that gave some of the characteristics to Chinese ethnic group or whatever. This idea was still floating in the air. So this is just a figure from Chris's early paper in 1974, where he pointed out that basically if you looked at this, if you tried to group things together according to their cranial morphology, it looked like this, that Neanderthals were a branch off in Europe, and then we've got African archaic and then moderns. Here's Chris actually here in Birmingham. We'll see him at the end of the lecture when he doesn't do a very good job of lip syncing to rap music. The things that I get people to do, you'll see. The fossil record actually is supportive of the out of Africa hypothesis because the earliest anatomically modern fossils are actually found in Africa. So the oldest ones we have are from Ethiopia, 195,000 years ago, and 150 to 160. And when you look outside of Africa for anatomically modern humans, what you see is much younger than that. So this is, in terms of just a fossil record, this holds, helps to sustain the out of Africa, recent African origins hypothesis. But it's from molecular phylogenetics that the hypothesis actually gained its most traction. And the early days, a couple of decades ago, where people started to look at this, looking at a number of different markers, particularly mitochondrial DNA. So the mitochondrion is a very unusual bacterium. It sometimes effectively functions as an organelle, but it is actually a bacterium. It has its own genome, and it's passed down on the maternal side. So it gives a nice, simple story about phylogenetics. The Y chromosome actually acts as an interesting counterpart because that's passed down the male lineage. So you can get a very quick and easy view of human lines of descent from looking at these two kind of markers. Problem is that when you look at these two markers, you do get a simple view, and you may end up with a slightly misleading view when you look at these alone. So the first attempts at mitochondrial analyses of human populations across the world came in the 1980s with this paper from Can et al. And this was the first time we came across this term mitochondrial Eve. Or African Eve, which is the ancestor of all existing mitochondrial genome sequences in humans. So these guys, they took 147 individuals. They didn't actually do sequencing, they just cut the DNA out with restriction enzymes, but they looked at the patterns, and they constructed a tree, and they rooted the tree in Africa about 200,000 years ago. So what they're saying is that all the mitochondrial DNAs that they had seen stem from one woman who lived in Africa 200,000 years ago. Now, there were some criticisms because, in fact, 18 out of the 20 African samples came from African Americans, and there was quite a large European admixture into African American populations. And people said, well, it's a bit misleading because using this biblical term of Eve makes it look as if she was the first human woman, but she wasn't. She was just part of a population. It just happens that her sequence got kind of fixed in human populations. A little while later, a better, more comprehensive analysis was done using complete mitochondrial DNA sequences, and this time they used quote-unquote real Africans, people actually coming from Africa directly. And what they found was that you could get a complete separation of the non-African lineages from the African lineages. So if you look at the top here, all of this here is all the variation you see outside of Africa, and you can see it's just one branch of a clade, which also has some African representatives, but there are many other deep branches in Africa, far deeper branches than what you see outside of Africa. So the most recent common ancestor for all of it was about 172,000 years ago,
but the most recent common ancestor for this main non-African branch was as short a time ago as 52,000 years ago. So this was good molecular evidence that all humans outside of Africa, so that pretty much everyone in this room here actually has African ancestry just a few tens of thousands of years ago. And when we look now, there's been much, much more work done. It's clear that this story is still true. It's borne out by these analyses. We've got these, all the non-African lineages derive from just two branches, the so-called M and N branches from the L3 haplotype of mitochondria. So if you look at the branch lengths, this is a very, very shallow branch of humans, even though geographically highly dispersed. So the yellow stuff over there is all the African stuff, and then all these other colours here, but you can see in Africa far more diversity. Y chromosome data said the same. So alongside mitochondrial Eve, the term Y Adam was coined, and again it's not the same as saying he was the only man or the first man or whatever, but that was placed about 60,000 years ago as well. And again, when you looked at the branching patterns, it was clear that Europeans and Asians, they were just one little branch branching off this much more diverse African tree. And this is a fairly recent review, basically says that all of the non-African lineages derive from one haplogroup, this CR haplogroup. So the same kind of pattern here that you've got these sub-Saharan Africa at one end here with very long branches, and then all of this geographical variation actually not reflected in very much in the way of sequence variation. And this is just a graphical display of the same kinds of patterns of these different mitochondrial and Y chromosome haplogroups over time. And you can see that they do map onto geography, but the actual branch lengths and so forth suggest that Africa was the origin of anatomically modern humans. And so now what we accept is that the peopling of the world happened from Africa. We had African origins 150,000, 200,000 years ago, and then a bit like with the chimpanzee, human chimpanzee, there's a little bit of variation. Some people say 70,000, some say 60,000, some go even as close as 50,000 years ago. But recently, humans left Africa and populated the world, and all the people outside of Africa are descendants of that migration out of Africa. And a very small number of, probably a small found population of only a few thousand individuals. And they populated the Eurasian continent. Interestingly, the earliest humans outside of Africa are found in Australia. So humans very quickly moved along into Australia, into Europe, into Asia, and then only relatively recently, about 15,000 years ago, maybe slightly earlier than that, maybe 20 to 25,000 years ago, moves into the Americas. But before we talk about variation outside of Africa, let's go back into Africa and think about what is the variation inside Africa, and what are the most deep branches within the human tree. Well, basically we have to focus on these individuals, the so-called San, or San people, otherwise known as Bushmen, hunter-gatherers from southern Africa, and they speak these Clegg languages. And this is when I went to South Africa a few years ago, I filmed this guy telling a story in Clegg language, and you can hear the click sound. You hear those distinctive sounds, the clicks that you make. And it turns out that these are the deepest branch here. So this is a paper that was trying to look at human matrilineal diversity on the mitochondrial side, and they found that there were 40, the deepest branch was this Khoisan lineage, which diversified from the rest of the human gene pool between 90 and 150,000 years ago,
And then there are about 40 other branches more recently than that. And so if you draw a phylogenetic tree based on the maternal, on the mitochondrial genomes, this is the sort of thing you get. So we're not, it's remarkable that there was so little diversity outside of Africa, but actually even inside Africa, all the people who aren't Bushmen are actually quite closely related when you consider the Khoisan over here, where the ancestry branched off really very close to the beginning of our species. And so tremendous, this variation that you see. So if we zoom in here, you can get some perspective there. So most of the people in this room, in this out of Africa clade over here, little out of Africa clade, these are all Africans in purple, other Africans in purple, but then over here you've got the Khoisan over there, with this huge divergence from the rest of humans in terms of. So you can just see that this L0 branch, the one that leads to the Khoisan, branched off such a long time before. That was based on mitochondria, but a few years ago, we for the first time actually got complete genomes, got complete nuclear genomes from Khoisan and from Bantus. So Bantus are another African group that live in southern Africa. They actually, many of them speak Click languages as well, but they stole the Clicks from the Bushmen. They probably actually migrated into that area relatively recently, and the Khoisan, or Bushmen, are the original inhabitants of the area. So what they did was they sequenced a number of different Khoisan individuals, these 4 men at the top here. Anyone know who that guy is? At the bottom there. Some of you might be heading that way. Tutu, Desmond Tutu. When I was a boy, we used to call Tutu a Desmond after this guy. Desmond Tutu, he's an Anglican archbishop in southern Africa, a very famous politician during the apartheid era. And his genome was sequenced as well as part of this initiative. And interestingly, I was actually in South Africa. I went to a conference on bioinformatics and heard the lead author of this paper describe these results while actually in South Africa. It was quite exciting. So we got these 4 individuals, and the remarkable thing was that when you looked at, say, 3 of these individuals and you counted the number of SNPs, single nucleotide polymorphisms, between these 3 different Bushmen from different parts of southern or southwest Africa, the distances between them were as great as they were between, say, a European and a Chinese. So there was as much variation in just that small part of Africa in these populations than there were in the whole of the Eurasian landmass. So really remarkable. So here they plotted on here. It says you've got on one side you've got the SNPs from one individual compared with a Yoruban and another Khoisan individual, and then on the other side here you've got that compared with Craig Venter as an example of European ancestry and a Han Chinese genome. So more variation among Bushmen than Eurasians. Interestingly, again, we have to be careful. The fact that they're so different is that they've actually accumulated lots of SNPs on their lineage, just the same as the rest of us have accumulated SNPs on our lineage. In fact, they seem to have accumulated more SNPs. And interestingly, when these genomes are sequenced, including Tutu's genome, who would have been classified as a Bantu, it's shown that there was actually gene flow. So basically, Desmond Tutu actually had a Bushman-type mitochondrial DNA haplotype, and one of the Bushmen actually had a Bantu-specific Y chromosome haplotype as well. So clearly these populations have been, to some degree, inbreeding. Now, in South Africa, politics of race was a predominant force when I was your age. The apartheid regime, it was a very difficult time, and then it all supposed to have gone away because the African National Congress took over. We had a majority government, and people didn't classify each other according to race anymore, except that they did, and the term coloured was still used as a term to deprecate people of mixed race, particularly in the Cape Province. The Zuma government said, there were too many colours in the Cape Province because they were voting for a non-ANC government in the Cape Province. And Tutu said, well, on the basis of his genome, he's coloured as well. It was kind of interesting to see how these genomes and all these racial things get mixed up into this politics at the time. Interestingly, the most basal Y chromosome haplotype comes from Africa, but not directly. It was found in an African American who submitted his genome, his DNA, to a phylogeny site, and they actually found this very, very deeply branching lineage. So that was a remarkable finding. Now, I'll have to move on a bit. I'm running out of time, aren't I? So if we look at the African exodus, then people probably left Africa from Ethiopia and through Eritrea into the Arabian Peninsula. Back in the day, there was a much smaller, narrower gap between the two continents, and the sea levels were a lot lower. And so it's quite conceivable that humans did actually make that jump. And then this idea of the beachcomber hypothesis, or the southern route, is that humans walked out of Africa and then just walked on the coast and found all their way down to Australia very quickly. And because a lot of that coast was actually, because of changes in sea level, you could actually just walk between these places that now are obscured by sea. And people have been looking at this more recently, looking at genomes of people in Ethiopia and Egypt, and showing that those individuals, what you'd expect is those populations in that corner of Africa, that's where the non-European or the non-African the populations originated, then they would show more similarity. And it turns out that they do. So there is some evidence that that is where humans left. I'm running out of time, so I'm not going to go into detail on this. It actually might not be quite so simple as I've made out. First Aboriginal genome sequence, in the paper there, they suggested maybe there were two different routes out of Africa. It wasn't just a single route. Maybe there was a route into Europe and Asia, and a separate route into Southern Asia and then into Australia. And I'm going to run out of that as well. So the interesting thing that we get now, going back to politics, if you like, just for a moment, is that if you look at the racial classification systems they use by the police force in America, I don't think even in this country perhaps, you've got this idea that there are these little pigeonholes you can put people in: African, Australian, Asian, European, American, and Oceanic. In fact, the view from modern genetics is much more complex than that, and that we've got these very deep branches where the Bushmen over here, and then the difference between a Native American, an Asian, and an Oceanian is minuscule in terms of the grand scheme of things. And do they really require separate racial classifications? And this, have you met Alice Roberts yet? She lectured you yet? I recruited her a few years ago when I was here. She did, for the Incredible Human Journey, she did a reconstruction of the first European made in black, which upset readers of the Daily Mail who didn't like the idea of it. But there we go. That's what the facts tell us. So, in conclusion, we're all only 4,000 mothers away from Africa. So whether you're the palest skinned Scandinavian or the darkest skinned Aborigine, all of these non-Africans are more closely related to each other than they are to any African. And all of us come from Africa in a relatively recent past. Okay, it's time for a little break. We'll have a video break now. So I'm now going to make a summary of this part of the lecture in a rap. I'm not going to do the rap. It's going to be done by a guy called Brinkman, who's a friend of mine, who I've known for about 10 years now. And I actually persuaded him in the run-up to the Darwin Bicentenary when I was writing the rough guide to evolution to produce the rap guide to evolution. And I said, I particularly want you to address the recent African origins theory in a rap song. Can you do that, please? And he said, yeah, I'll have a go. And this is what he produced. And in fact, the video was filmed just over in the next building along in chemistry
here in Birmingham.
Yeah, Africa. I'm an African, yeah, and I know what's happening. I'm an African, archaeologists know what's happening. You an African, yeah. Do you know what's happening? I'm an African, geneticists know what's happening. No, I wasn't born in Ghana, but Africa's my mama, cause that's where my mama got her mitochondria. You can try to fight if you wanna, but it's not gonna change me, cause it's plain to see, Africans are my people. And if it's not plain to see, then your eyes deceive you. I'm talking primeval. The DNA of my veins tells a story that reasonable people find believable, but it might even blow your transistors. Africa is the home of our most recent common ancestors, which means human beings are all brothers and sisters. To check the massive evidence of Homo erectus and Australopithecus afarensis in the fossil record, and then try to tell me that we're not all connected. The fossil record has gaps but no contradictions, and it complements the evidence in your chromosomes. So I came to let you know about your ancestral home. I'm an African, yeah, and I know what's happening. I'm an African, archaeologists know what's happening. You an African, yeah. Do you know what's happening? I'm an African, geneticists know what's happening. Yeah, it's plain to see. You can't change me, cause I'ma be a Homo sapien for life. Yeah, it's plain to see. You can't change me, cause I'ma be a Homo sapien for life. Yeah, the red is for the blood in my arm. It runs in the veins of all my cousins from the same African mom. And the black is for the melanin, which I guess I lost. A mutation with benefits that offset the cost, at least in the north. After massive glaciation, my family passed through some adaptive radiations. We started as Africans, and then became Eurasians. And then one final migration made us Canadians. But it's back to my origin, cause I understand for every color of man, Africa's the motherland. So I'm coming back, that's my right of return. I'm only speaking the facts, which I invite you to learn. We came from Africa first, Charles Darwin predicted it, cause that's where modern chimps and gorillas live. So the green is for the envy in the eyes of intelligent design advocates and scientific illiterates. I'm an African, yeah, and I know what's happening. I'm an African, archaeologists know what's happening. You an African, yeah. Do you know what's happening? I'm an African, geneticists know what's happening. Yeah, CANADA, Asia, UK, USA, RUSSIA. No, it ain't about where you stay, it's about the motherland. CHINAAUSTRIA, Oz, Tasmania, and Ukraine. No, it ain't about where you stay, it's about the motherland. MONGOLIA, Saudi Arabia and Kuwait, Sweden, Denmark, and Norway. No, it ain't about where you stay, it's about the motherland. COLOMBIA, Costa Rica, INDIA, First Nations, and ALASKA. No, it ain't about where you stay, it's about the motherland.
Right, now we have to wait. Oh, I'll take that off. The end. You think it's all over? About African, the fact that Pan-Africanism. But anyway, you think it's finished, and then suddenly he goes on and collects this video from Chris Stream.
I'm an African, and I know what's happening.
That'll be perfect. Do it one more time.
I'm an African, and I know what's happening.
That's it.
So that's the author of the Out of Africa hypothesis. I managed to get him to do that wooden delivery out there in Chancellor's Court. And we decided, Baba made a good choice, I think, not to mix him in, but give him his own spot at the end there. So I've told you a bit about genes and our most, our closest living relatives. Told you about the Out of Africa hypothesis. But what about actually looking into the past? We'll start with a particular conundrum. Start in a place called Neanderthal, which is a valley near Düsseldorf in Germany. And for some reason, it was named after a local pastor called Neumann, but they turned it into Greek. Neander just means Newman in Greek. And so the valley was named after him, and Tal just means valley. I think it's cognated Dale in English. But they changed their spelling in 1901, and unfortunately, you see Neanderthal with and without the H used in scientific papers
And if you go and search PubMed, you have to search with both because they don't alias them to each other. Science uses without the H, nature uses with the H. It's one of those frustrations. But anyway, I'm getting ahead of myself here. What happened was that there were lots of caves along this particular valley, and limestone quarrying began with the industrialisation of Germany, and they started knocking down all the caves and digging them up. And in one of these caves, they discovered a skull cap and some postcranial bones, and they were identified by a local teacher and historian who said, these aren't a bear, which is what people thought, these are actually some kind of archaic human. And so they're written up in a paper in 1857, just at the right time for when Darwin's theory of evolution came along a couple of years later to actually dovetail nicely. One of the most interesting stories, though, in paleoanthropology is that site was left for 150 years, and then people went back around the turn of the millennium and said, oh, see if we can find where the original site was, and it had been obscured because there had been so much quarrying going on. But using various landmarks, they found it, they dug down, and they found more Neanderthal remains. And some of those remains, one bone in particular, fitted like pieces of a jigsaw into one of the original bones found 150 years earlier. So I think that's just a truly remarkable story of looking at the type site. So this is what the valley used to look like, kind of, a bit like Cheddar Gorge. This is what it looks like now. So those poles there are the site where those most recent bones were found. The original site is to one side here. It's probably a bit of an escarpment there where the cave was, but it's all just been excavated away now. It's all quite flat around the river. It's a quite peaceful place, actually, in the countryside near Dusseldorf. These are my own photographs. In the century and a half since those discoveries, there's now been over 400 specimens of Neanderthal man have been found. It's actually one of the it's probably better documented, in fact, than anatomically modern humans over that particular time scale. And these samples have been found all across Europe, at least the parts that were not covered by glacial sheets, and more recently it's been found in parts of Asia as well, and into Siberia. We found Neanderthals. And the reconstructions have varied. So this kind of brutish looking thing here is found outside the Neanderthal Museum, the Neanderthal site, and that was the early view. These were brutish cavemen. More recently there have been much more sympathetic reconstructions to show that these individuals actually are not that different from modern humans, anatomically modern humans. I particularly like this little girl here, very sympathetic reconstruction there of a Neanderthal child. And in fact, in the museum, they actually reconstruct a Neanderthal man, and they put him in a suit to show that if you were walking down the street in Birmingham, down Broad Street, you wouldn't notice. You wouldn't see anything different. You may disagree, but we actually have an age-matched, sex-matched control here, and you can see there is quite a lot of similarities between anatomically modern humans, particularly Europeans, and Neanderthals. So Neanderthal features first seen about 350,000 years ago, and then the full-blown Neanderthals were thought to have lived from about 130,000 to 40,000 years ago. And they overlapped with anatomically modern humans for at least 5,000 years in Europe. Now, we have no evidence of them actually living together on the same site, any fossil site, but they kind of overlapped in terms of their geography in Europe and West Asia. And so that raises a question. But before we go there, it's worth stressing that although I've made out that they are very similar to us, they also were distinctive in certain ways, anatomically. There were probably more differences between us and Neanderthals than there was between the common chimp and the bonobo. Much more robust than we are. They have a pronounced brow ridge, projecting mid-face, kind of chinless, but they have, in fact, larger brains on average than modern humans. So the question was, was there a genetic exchange between these anatomically modern humans coming out of Africa and Neanderthals? So there we are, we march out of Africa into Eurasia. Did we meet Neanderthals and mate with them? Did we kill them and wipe them out? Or were we indifferent to them? So the first studies on this were done in the 1990s, where Svante Pääbo, who was one of the godfathers of this field of paleoanthropology, of DNA sequences, showed that basically the sequences of Neanderthals in their mitochondria were outside of the range of modern humans. And so this basically spoke to the idea that this is a separate species. They went off their way, modern humans went off our way, and we never met, and that's it. And subsequent papers up into the new millennium started saying the same thing when you looked at more mitochondrial sequences and got longer sequences. And the evidence for mitochondrial DNA was basically they're different. And this means two separate species, no admixture. So here are all the different sequences that were obtained by 2007, and you can see that they all have common differences that are different from the so-called Cambridge reference sequence, the anatomically modern sequence. But then in 2006, the world changed because suddenly we got paleogenomics. Svante Pääbo managed to get genome-level sequences out of a Neanderthal. And we first got 1 million bases. We got two separate papers in parallel, one of which said 1 million bases. I think the other one was 62,000. Both of these papers had the potential to change our way of thinking because suddenly we were getting much more information. But both papers kind of hedged their bets and said, well, we can't rule out, we can't rule out very small amounts of admixture. There's no large amounts of admixture, but it looks as if there is really not much evidence for it. So certainly there's nothing there to. So basically the confidence estimate ranges from 0 to 20%. So a definitive answer will require additional sequence data. Additional sequence data came along in 2010 with a first draft sequence of the Neanderthal genome. And what they showed in this draft sequence of the Neanderthal genome was that they compared it with anatomically modern humans, and they said, well, is it, are Neanderthals more closely related to any other? If they just branched off, they would be equidistant from all modern humans. But maybe they're more closely related to Europeans. Let's have a look. So they compared with Europeans, East Asians, and West Africans. And what they found, surprisingly, was that not the Neanderthals were closely related only to Europeans. Neanderthals actually were closely related to all non-Africans. And then they confirmed this by looking at more genomes, and they found that there was a direction of flow was that actually Neanderthals gave some sequences to non-African humans. So it was interesting to note that Craig Venter, whose genome was the definitive genome at the time, there were stretches of his genome that were more Neanderthal than they were African. And the takeaway figure from this paper was somewhere between 1% and 4% of non-African genomes are from Neanderthals. And so when we looked at the human family tree, the phylogenetic tree, what we see is with anatomically modern humans, we've got the sand branching off very early, we've got West Africans, and then Papuans, Chinese and Europeans, all the non-Africans there. Neanderthals, there was an infusion of Neanderthal sequences into that branch there, in that out of Africa branch. Now, this is so commonplace, you can just spit in a tube, as I have done, send your DNA off to 23andMe, and they'll tell you how much Neanderthal ancestry you have. So I can tell you that I have 2.8% Neanderthal ancestry. The average 23andMe user is 2.7. My daughter is 2.7. Suggests that my wife might even be less Neanderthal than that. And you can actually buy a Neanderthal and Proud t-shirt if you send your DNA off to 23andMe. And you can see, example, a Japanese person has 2.7, a Chinese person has 2.5, but if you're Nigerian, 0.3% chance of 0.3% Neanderthal ancestry. So one of the things that people started doing once they got the Neanderthal sequences was saying, same as we said with the chimpanzee variation, well, is any of this variation between the sequences biologically meaningful? And they found that there was an allele in a particular receptor, a melanocortin receptor, that actually might be associated with pigmentation in Neanderthals. And Neanderthals probably were pale-skinned and freckled because of what they've been finding here. And there's been a whole number of other papers as well showing the same kind of things that showed that FOXP2, the variant we have, that's in this so-called language gene, Neanderthals had it too. And in recent years, there's been a lot of interest in actually mapping out the Neanderthal ancestry in present-day humans. And it turns out that 2.7% is not just smeared out randomly across all the chromosomes. There are parts of the genome where there's large amounts of Neanderthal ancestry in our non-African populations, and other parts where there are Neanderthal deserts, if you like, where Neanderthal sequences have been selected against. And there's been a lot of interest in actually trying to resurrect Neanderthal lineages from modern humans. And so they're now suggesting that they can recover about 20% of the Neanderthal genome just from modern genomes. So this is a remarkable finding. How am I doing? I'm going to run out of time, but we've got more to come. The best is yet to come. So the best is that I was blown away. I used to teach this course. I started teaching this course in 2006, 2007. It was just like a roller coaster. Every year there would be new findings. Bloody hell, we got a Neanderthal sequence. Then we found something even more remarkable. So from a finger bone found in a cave in Siberia, they got a mitochondrial genome sequence. The mitochondrial genome sequence they got didn't look like anything they'd seen before. It looked outside of the variation seen in modern humans, but even outside the variation seen in Neanderthals. And so this so-called Denisovan genome, in this Denisova cave, is out there as an outgroup. So you've got, that's all the humans up the top, anatomically modern humans. The blue bit is the Neanderthals. The red stuff is this Denisovan genome. So this is found in this cave right in the middle of the Eurasian landmass there in Siberia. And it just keeps motoring on this field. They then actually got a genome, nuclear genome, 1.9-fold genome, and showed that basically this archaic hominin group, the Denisovans, were more closely related to the Neanderthals than they were to us, but they had actually contributed a certain percentage of the genome to some modern human groups, particularly Melanesians. So people from Papua New Guinea, and it turns out also Australian Aborigines have an admixture from this Denisovan group. And so the Denisovans or Denisovans, they also play their role in this human phylogeny. They actually diverged early in the Neanderthals, but then actually in a very deep branch of the human phylogeny, the branch leading to the Papua New Guineans and the Aborigines, they gave some DNA. And we've now got high coverage sequence, 30-fold. And one of the things that's happening, I'm running out of time, is that people are now looking at modern human genomes and saying, well, which parts of the genome actually come from African ancestry, which parts are the result of introgression from these other archaic lineages, and how does that actually have an effect on biology? And this is a really fast-moving field. Lots of interest here in looking at the phenotypic legacy of admixture between humans and Neanderthals, Denisovans as well in terms of immune systems. And you might have seen, it's even made it to the Daily Mail recently where they said Inuit people have the same caveman genes that helped an extinctive human survive. So basically, that they've shown that Inuit actually seem to have some admixture from the Denisovans as well. So the top figure there just shows you modern humans came out of Africa, they met the Neanderthals, and somehow they met the Denisovans as well. It's still a mystery because the Siberian cave is a long way from the kind of migration route that people reconstructed going out to Australia and Papua New Guinea, but somehow the Denisovans met modern humans along the way. And this figure here also shows you that some parts of the genome you've got a lot of Neanderthal in the European, other parts you've got much less. It's not just random. Anyway, I'm losing my audience because you're running out of time. There's a thing about amylase. So basically, all I said to you about amylase earlier, about that was a change between us and chimpanzees. In fact, that was a change between us and Neanderthals. So that is an interesting finding. And on it goes. This lady asking about epigenetics earlier. They've actually done epigenetic reconstructions of the differences between Neanderthals, Denisovans, and modern humans. And recently there was a paper where they looked at a human from Romania, an anatomically modern human from Romania, looked at the genome and found that that individual actually had such strong admixture from the Neanderthal that Neanderthal must have been a great-great-grandmother or great-great-grandfather. So basically the relationship between Queen Elizabeth and Queen Victoria, this individual, and these green bars are bits of the genome that are actually Neanderthal admixture. Almost finished now, just a few minutes left, but nearly done. So last year, about a year ago in fact, I went to this place, Atapuerca, in northern Spain, which is the site of the earliest hominin remains in Western Europe. And it's a remarkable site. They've actually got this cave, and they've got this thing called the Cima de los Huesos, which is like this pit within the cave structure where bones have just fallen down this big shaft and accumulated there. And the paleoanthropologists there have discovered a new, what they've called it Homo antecessor, a new variety of humans that has some Neanderthal features, some anatomically modern features, maybe close to the ancestor, the common ancestor of humans and Neanderthals. But the strange thing was when the mitochondrial genome was sequenced for this, it turned out to be more like a Denisovan than the Neanderthal, which is a very strange finding. But when I was out there last year, I said, "Oh, have you not got a nuclear genome yet? Haven't you solved this problem?" And they kept saying, "We cannot tell you, we'd have to kill you. It's under embargo at Nature. We can't tell you." 2 weeks later, when I got back, we got nuclear sequences, which showed that actually the nuclear genome of these individuals was more like Neanderthals than Denisovans. So basically there was admixture going on between lots of different lineages over time, and this is getting a much more murky and complicated field as time goes on. The number of different admixture events that have gone on from different lineages over time being reconstructed is growing all the time. So there's now a potential unknown hominin lineage where people have just detected this admixture without any fossils and don't know what the things look like, where they lived or anything like that. And that's probably going to get worse, probably going to get some more of those. Anyway, I think I've just about finished. If you're interested in it, if any of you are particularly interested in Pete and you want something to read that's fun, you can get this book Neanderthal Man by Svante Pääbo. Svante Pääbo is just one of those remarkable guys who basically jump-started this whole field. He's the one who invented ancient human DNA as a study, the field of study. And he's written a memoir recently talking about his experiences in the field and how he progressively moved forward. But the other interesting thing is he leaves all sorts of weird details about his personal life, about his sexuality and all sorts of other things in there as well. So it's kind of interesting. And if you want to see Alice in all her glory, she did do a documentary, I don't know if anyone saw it, a few months ago. It was on BBC, Horizon, actually looking at all this stuff about archaic human lineages and so forth. And it is available it's not on YouTube because I keep taking it down, so there's copyright things. But I did find one site that had a copy of the video, if you're interested. It's probably still there. So in conclusion, we've got multiple Neanderthal mitochondrial sequences, and they suggested early on that there was no admixture, that it was a completely separate species. But now we have genomes, we know that there was this small-scale mixing, we know that there was mixing from Denisovans and other populations, and that now people are picking apart the biological consequences of this, and it's just going to get more and more. There's going to be more and more gene and genome sequences coming out of this field in the future. And now I'm going to finish with Alice. Alice can't sing in real life, but they made her sing in this video. And we'll just watch this as a summary of the whole 2-hour show, and then you can go home. Oh, wait a minute, that's not right. There we go.
Man is a singular creature. He has certain gifts which make him unique among the animals, so that, unlike them, he is not a figure in the landscape. He is the shape of the landscape.
We are children of Africa. Children of Africa. They say this is where it all began. This is where it all began.
In a parched African landscape, man first put his foot to the ground, to the ground, to the ground, to the ground.
Africa was our only home for tens of thousands of years, until a small handful of people made their way out of Africa. These beings with soaring imagination, and eventually flung themselves and their machines into interplanetary space. These beings with soaring imagination, eventually flung themselves and their machines into interplanetary space. We are children of Africa. Children of Africa. This landscape has been home to humans 200,000 years. We have come so far. All of this is cause for great celebration. We've come so far. This is a story about us. Those early Europeans were people like you and me. But it is humbling when you see the challenges they faced. People like you and me overcame the Neanderthals. People like you and me made it through the Ice Age. These beings with soaring imagination, eventually flung themselves and their machines into interplanetary space. We are not the only beings with personalities, minds and feelings. Chimpanzees have very clear personalities, minds and feelings.
Take a chimp and feed it with me, and let it go 2 or 3 more rounds of division. Out comes symphonies and ideology.
We are not the only beings.
Everything that we are that distinguishes us from chimps emerges from that 1% difference in DNA.
Like you and me overcame the Neanderthals. Like you and me made it through the Ice Age. These beings with soaring imagination, eventually flung themselves and their machines into interplanetary space. These beings with soaring imagination, eventually flung themselves and their machines into interplanetary space.
Using his burgeoning intelligence, this most successful of all mammals has exploited the environment to produce food for an ever-increasing population. Instead of controlling the environment for the benefit of the population, perhaps it's time we control the population to allow the survival of the environment.
Yeah. Good. Thank you very much then. So that's me finished. Happy to take any questions.